Hello, everyone. My name is Justin Baker. This is the fourth lesson in my biology's fourth unit on and we're going to talk about genetics. We're close to wrapping up this unit, and we're going to talk about karyotypes today. We're specifically going to ask the question, how are karyotypes used to study genetic disorders? All right, so karyotypes, just right off the bat, it's important to know what they are. They're an image of the homologous chromosomes generally arranged in decreasing size. All right, so we've got sister chromatids that are homologous, right? And they're going to be arranged in decreasing size. So humans are diploid. Most things are. That means they have two copies, right? So if we look at our example karyotype here, and this one is just black and white. A lot of times these are dyed specifically so that you can see different things on it. This one, just black and white. We're keeping it simple. So you can see how there are different sizes. Most chromosomes are going to be in this X shape because, remember, they're joined at the centromeres, right, here. And so we've actually got two sister chromatids, two sister chromatids, right? And they're together as homologous chromosomes. Humans have 22 of these plus the 23rd, which we call the sex chromosomes. And these are either going to be completely paired or they're going to be slightly different. And so we've got female sex and we've got male sex chromosomes. Female is two X's, males an X and a Y. That's what a karyotype is. That's all it is. So these are usually dyed so that we can see specific things on them, all right? So they're going to look a little different. They may be idealized maps as well. So they're going to be like perfect images instead of ones that look like this. Right? And you can actually kind of see this architecture here on the side. That is the banding that we've talked about before when we talked about gene mapping. Right? So the big problem here with genetic testing is looking for non-disjunction. Now, non-disjunction is when, during meiotic division, something goes wrong. The disjunction goes wrong, right? So non-disjunction means it doesn't happen. No disjunction. So during meiosis 1, it can happen, or during meiosis 2, it can happen. So on this side, that is what we're supposed to look like. If you remember meiosis, this is exactly what it was. So we had the parent cell, right? And it would double its chromosomes, and they would split apart, right? So during meiosis 1, they would be split apart. During meiosis 2, those get split apart again, right? To make gametes. Gametes are our sex cells, our germ cells. And so I can't put my cursor down there because it's going to bring up the Google Slides menu, but in the corner opposite my face, so way down there, right, we have perfect gametes where each gamete looks exactly like the other gametes in terms of the amount of chromosomes or chromatids that it has. So when they join together through pairings, right, we would get double that, right? These are haploid. They are haploid, single sets. Non-disjunction during meiosis 1 means that one of the daughter cells that goes into meiosis 2 has more than the other, right? And it's going to have a lot more than the other, right? So this one, in our example here, gets both of these paired chromosomes, right? These homologous chromosomes that are here, they move to one side instead of splitting to both sides, right? Where non-disjunction in meiosis 2, that works okay, but in meiosis 2, they don't split apart like they're supposed to, right? So this one is what's supposed to happen. This one is what's not supposed to happen. So we get one gamete that has an extra chromosome, we would say, or an extra chromatid, technically, where this one has one less chromatid, and these are okay. For meiosis 2, or if it's non-disjunction in meiosis 1, we get 50% of our gametes that have an extra and 50% that have a missing one. If it's in meiosis 2, we have 25% with extra and 25% without. Now, a lot of times, non-disjunction results in a fatality, all right? It doesn't result in a successful pregnancy, let's say. There's no good way to say that, right? But sometimes the offspring is still viable, right? And this leads us to a few different disorders, right? But karyotype genetic studying is mainly done to check to make sure that an offspring has the correct number of all of its genes, right? That's it. Now, a more in-depth karyotyping may be done to see which chromosomes a offspring has inherited from the parents, right? Once again, we're scientists. We don't care if it's good. We don't care if it's bad. We're just saying this is a thing that happens, right? And that testing can be done to predict what a phenotype will look like for an offspring, right? So we're going to continue studying karyotypes this week in my class. I know this is pretty simple, but it does get complicated and it gets kind of into some moral gray areas as well. But karyotypes are used to study genetic orders by looking at which chromosomes a offspring has. Right? As always, keep asking questions, keep learning.